Hello everyone, welcome to Biovidya. In this video, we are going to introduce industrial microbiology and learn some basic concepts of it. What is industrial microbiology? In simple words, we can define it as industrial application of microbes. But if we define it properly, it is a branch of science that deals with biotechnological aspects of using microorganisms for large-scale production or overproduction of various goods of human interest from comparatively inexpensive or cheap raw materials. Biotechnology and industrial microbiology are linked together, and in biotechnology, microorganisms are genetically engineered in such a way that they are able to produce the product of interest or substances that they would otherwise not able to produce in normal circumstances. And in industrial microbiology, the use of those genetically engineered microorganisms for their overproduction. And to achieve this or doing so, microorganisms are grown in controlled environments with specific limitations to maximize the synthesis of the desired products. And potent microorganisms are those that can convert inexpensive raw materials into economically valuable products. Now, major discoveries. It originated with alcoholic fermentation process like beer and wine. In 1886, first pure culture of yeast was used. Large-scale production of glycerol during the First World War. The penicillin, which is an antibiotic, which is industrially produced during the Second World War and which is considered as a landmark in industrial microbiology. Amino acid fermentation in the form of L-glutamic acid was industrialized in 1957. SCPs, the single cell proteins, were industrially produced in late 1980s. Now we have to know about what are primary and secondary metabolites. Primary metabolites are the compounds that are used in the synthesis of microbial cell and are produced in the growth phase, that is the trophophase. These are essential compounds for cell's growth and development, and they are synthesized during the active growth phase. Now, the examples are amino acids, nucleotides, and fermentation end products. The secondary metabolites are not essential for the cell's growth and development, and they have no direct relationship to the synthesis of the cell's material and normal growth, and they accumulate during the EDO phase, that is the period of nutrient deficit or limitation or waste product accumulation time, that follows just after the active growth phase, and antibiotics and mycotoxins are the examples. Now if we see bacterial growth curve, it is somewhat like that, where this is the lag phase, this is the log phase or active growth phase, this is the stationary phase, and this is the death phase. Primary metabolites are synthesized along the active growth phase, and secondary metabolites are synthesized just after the active growth phase, that is the early stationary phase, and are mainly accumulated during the late stationary phase or the death phase. Now, what are the criteria for being a potent industrial microorganism? First, they should have rapid growth potential. They should produce spores or some other reproductive cell structures in huge number so that they can be used as inoculum for large scale production. Next, they must have the ability to produce desired product in a relatively short period of time, that is, they must be time efficient. They must be able to grow in a liquid culture medium so that they can be obtainable in bulk quantities at a low price that make them cost effective. The organism must be capable of growth and product formation in large scale culture. They must be non-pathogenic, that is, they must not be pathogenic, especially to the humans or other economically important animals and plants. The microorganism should be amenable to genetic analysis because the yields necessary to make an industrial process profitable. Now, what are the processes involved in developing a potent microorganism strain? First, they are isolated and screened. Now, the isolated and screened microorganisms generally have a low yield or have a low production of the desired product and are unsuitable for the industrial application. Yield can be increased to a certain level by making the variations in the growth media and the physiochemical composition of the media. Change in the genetic constitution induced by mutagenic agents being the most effective factor for making a microorganism potent for industrial scale. The growth media of successfully genetically manipulated strain can be standardized. Now the modified strain can be used if it fulfills the above mentioned criteria. Now the strain is tested for its both qualitative and quantitative production in the laboratory scale fermenter. Next, to avoid any changes in the desirable products or properties by spontaneous mutation of the microorganism, it is preserved by freeze drying or in liquid nitrogen. Now, about major microbial products and the producing organisms, there are industrial products, there are agricultural products, there are food additives, there are medical products, as well as there are biofuels, and that this is the list of various major industrial products and the microorganisms involved in it. Now, the scopes of industrial microbiology. The microbial world is vast and highly diverse, and they possess metabolic abilities to produce great varieties of substances. The prospects of utilizing such substances for human benefit are practically unlimited. However, comparatively very few of those possibilities have been explored for industrial microbiology till now. And some of them are bioprospecting, which is the uncultured microorganisms world, and it is the major challenge in biotechnology to be able to explore those microorganisms which has not been cultured till now. Natural genetic engineering or forced evolution and adaptive mutations which are involved in developing a microorganism strain to make it industrially beneficial and commercially beneficial. Microbial growth in controlled environment is very expensive. Development of growth media is of much importance, and specific growth media and growth media composition and growth conditions for the growth of the microorganisms in a cost effective way is a large part of the industrial microbiology and biotechnology. Biosensors, the streptavidin biotin linked system for essentially real time detection of important pathogens, has been developed. So there are a lot of areas to be explored in industrial microbiology for humankind for a better future.